mitosis and the cell cycle. There will be some drawing tasks that you will do in the middle of this, and then some writing that you will do. I would set yourself up with like a T-chart, so somewhere where you can do all the drawing, and then a column where you can write the text in here. Overview before we get going is this is essentially what we are talking about, and that is how does a cell make another copy of itself? So you started with one cell. We're going to inevitably need more than one cell at the end of the process. This is how you get two cells at the end of this cycle. So remember we talked about these things in class on Monday. Why in the heck do cells need to divide in the first place? What do you need to replicate your DNA for? These are a couple of the reasons here. We're going to replace damaged or dead cells if you've gotten, say, a boo-boo. And also for growth. So you are not the same 6-pound baby that you were when you were born. You are a much larger individual now, and it is thanks to this process that you are able to grow up and be the person that I get to teach in class these days. First things first is that a cell doesn't do this all the time. This is not a continual process that's just happening every moment of the day. It's on a excuse me, timer system, I guess you could say. It takes a thing called a growth factor, and there are multiple different types of growth factors. These are 8 of them. You can pause the video here and read a little bit about each one of those growth factors. But one of those little growth factors needs to go and it needs to tap the cell and it needs to say, hey, we need you to make another copy of yourself. We need 2 of you. When you're little, you have lots of growth factor hormones. This is a type of hormone that is floating around in your body and is getting you to grow. And as you age, your growth factors slow down. They never stop. You just have a lot of them at various periods of time in your life. And then you slow down their release and their construction and their work that they do later on in life. So this process that we're going to talk about here only has to do with a thing called a somatic cell or your body cell. So these are going to be your muscles and your liver and your pancreas and your bones and your bladder and your skin and all of those regular body cells, everything that makes up your body. There is one other special kind of cell. We're going to deal with that later. I need you to lock the word body and somatic in your brain because those are the same thing. One is the fancy term and the other is the non-fancy version of the word. This is the other cell type. We'll give you a quick little preview here of this, but really it's not going to come into play until a later unit. But this is the other type of division that happens. It's called meiosis. We'll do an entire, I will lecture and we'll color all together with that. These are called your sex cells or your germ cells are the fancy way to say that. And you can see that they're different between men and women. So this is how you make babies at the end. Before you can make babies, you have to have these types of cells prepped and ready to go. So 2 different cell lines that we were talking about, and the process is incredibly similar, but it is different for these 2 cell lines. For this video, we're talking somatic cells. So we're just going to talk about your general, regular, run-of-the-mill body cells. This is essentially the process as it happens. It is the same picture that you have on the front of the activity that you did on Monday night. So on the front of the cell cycle activity, you have this picture. You are welcome to just annotate this picture. You don't have to redraw it, but you are more than welcome to redraw it. Cell cycle happens in really 2 parts. We call the first part interphase and the second part mitosis. Within interphase, what you focused a lot in that activity on was what happens during G1, what happens during S, and then what happens during G2. And you can see them labeled in the green section over to the side here. On your diagram, you're going to want to add a nice large arrow in the middle that says interphase. Or if you're drawing this, then you are drawing all of it already. Interphase is missing off of the activity that I had you complete. So you'll need to add that one. But from the table on the inside, most of you nailed this. I was peeking over your shoulder. But during G1, you have cell growth. During S phase, S stands for synthesis. The DNA gets copied, and that's the activity that we did where we replicated DNA. And the video that we watched about how Mendelssohn and Stahl figured that out. So that's where that happens during S phase. And then during G2, there is growth and divisions. We do a little more, no division, prep for division. We do a little more growing, really just like make sure that everything is going along according to plan. And then if everything has gone along according to plan, we get to go through the last two pieces of the cycle that we'll talk about in a little bit here. Would you sketch this picture for yourself? This is the interphase picture that I would like you to draw. So hit the pause button, take what you need to draw that picture, and then resume the video. For interphase, but I would like you to write down right next to it. These are the big highlights that I really want you to have and take with you. If you totaled all the time up that is on the table on the inside, you would see that it is pretty darn close to 20 hours. And that time, those are averages. Most of the cell's time during this divisional process is spent in interphase. This is much like painting a house or a room. If you have ever experienced that, you know that most of the time you spend prepping for the painting process. The actual paint on the wall doesn't take quite as long. It's the fun part, but it's not the part that takes very long. G1, again, growth, if you missed that, or you would like to label it on the front of your diagram. S is for synthesis of the DNA. G2 is for division. We're just prepping for that division. And then you guys also had on your packet a G0 part. There are just some cells that just never go through G0. So those of you that labeled somewhere in between, and this diagram is just on a different angle than yours is, somewhere between cytokinesis and GAP1, you put G0, you nailed it. There are just some cells that simply never divide. You get new ones. You can make new red blood cells. You're constantly making new white blood cells. You do replace neurons if they get damaged. You do replace other sorts of muscle cells. If your muscle gets weak, let's say you have to put it in a cast and then you get it back, you do make new ones. These three cells just never go through the meiotic process. So they just do not replace themselves via mitosis. They replace themselves. It's just a different process to get a new one. They can't go through this exact process. And we are not going to talk about that. It's also why if you lose a portion of any one of these cells, you can't get them back. If you lose too much blood, you have to actually get a blood transfusion. If your body stops producing white blood cells, you have to get a bone marrow transplant. If your muscles atrophy, you're going to spend a lot of time in an occupational therapy program, or you might even go to an inpatient hospital program where you have to literally build that muscle back again. And in some parts, you just will never get it back. It's too far gone. There's no originating cell from which to produce new stuff. Nerve cells, those are your brain cells. And this is why things like traumatic brain injuries are such a big deal, because once you lose that brain cell, you have to start all over. That's why people who lose the ability to walk have to start all over from the beginning, learning how to walk. If you damage your spinal cord, that's a nerve cell. You might forever be paralyzed, and you just don't have the ability to do certain things. Hopefully, we can retrain your body and get you a new copy, but you'll never go through this process that we're going to talk about. It just doesn't happen with these cell lines. The best part, the painting of the room, takes place during M phase and cytokinesis. Both parts are equally important, but they do not take up the same amount of time to go through. So hit the pause button, and I want you to draw out what happens in prophase. Yes, you have this in your textbook, so you can corroborate everything that I am talking to you about right now, but you need to draw it. Drawing will help it to stick in your brain. It will actually help it to make its way in there so that you understand and you know what the heck this process is. So Prophase. Chromatin is going to condense to become a chromosome. If you are going, okay, cool, whatever, I just wrote that down. Hit the pause button on that thought, and we're going to do it in the next slide. Talk a little more about chromatin and a chromosome. It is also why in this you have some extra pieces that are there, and you drew that the nuclear membrane is disappearing, and you have X's in this picture. But back here in this picture, all the way back, you don't have X's. You do not have X's here. We'll talk about why. You do have X's here, and we'll talk about why. You're going to lose your nuclear membrane. Remember, we've gone through S phase, so we have synthesized a brand new copy of the DNA. That means we have now 6.4 billion base pairs. We had 3.2, now we're up to 6.4. So it's just too much genetic information to be able to hold in that cell membrane, that nuclear membrane. It's just too much information. So it's going to disintegrate. We also have to do a couple things. We need it to move out of the way. You get a brand new organelle. So one that we haven't talked about or seen before, but you get this thing called a centriole. And centrioles are, I think of them like the Spider-Man of the organelles. They will literally shoot out webs from them. We'll talk more about why you have an organelle. It's a little part of your cell that can shoot out and act like Spider-Man. It's pretty cool. The things that it shoots out are called spindle fibers, and they have a very important purpose. But for now, I just need you to know that these things are appearing and they're going to shoot out the web. The web has a name. It's called a spindle fiber. Back to chromatin and chromosome. So this is why you had a scribbly thing and then an X. Yes, please hit the pause button. Scribble those two things, draw those things out so that you have them. And here's the difference. One is just the condensed version of the other. They're both DNA. It came out of the nucleus, therefore it has to be DNA. The uncondensed version we call chromatin, and it's not disorganized, it's just not hyper condensed up. When we end up and we take it from 3.2 to 6.4 billion base pairs, we just have to be incredibly detailed and organized. So then we condense things down into these things called chromosomes. Chromatin, loose form, chromosome, super condensed form. You can think of it like the extension cord. There's parts that are in that extension cord. It's not disorganized. You can untangle it. You can easily use the extension cord in that state. It's just not hyper organized. So you can see in the bottom little picture that if you wrap everything up, then it's hyper organized. Exact same thing here. Chromatin, chromosome, same thing, both DNA, just two different forms. And from here on out, we're sticking with chromosomes. We'll go back to chromatin, but not yet. So pause button again. Please draw this out for metaphase. You can see those spindle fibers are a little more prominent now. And we got some other things that are here. Yes, the extra picture that you have drawn over on the side, that I have drawn over on the side, yes, you need to do that as well. Please draw that guy, both of them. Okay, metaphase. I think of metaphase like middle. Starts with an M, and look what's showing in the picture, and the one you just drew, is that we're putting those chromosomes that were condensed in prophase down the middle of the cell. Okay? To get them to go to the middle of the cell and to make sure that they stay exactly in their spot, we are going to shoot out from the centromere that spindle fiber, and it is going to stick onto the chromosome. It sticks at a point on the chromosome. The X point where it actually sticks together is a bundle of proteins, and it's almost like a magnet. You can think of it like, I don't know, something that's very, very sticky, or magnet is a good example, or Velcro would be one, but it sticks on there. So the spindle fiber is going to stick to that centromere. Pause button again. Next phase, anaphase. So prophase, we have prepped everything. Metaphase, we lined it up down the middle. And now here in anaphase, we are going to move those chromosomes away from one another. Please keep in mind that, remember, we are talking about a duplicate set of your DNA. So you have two copies of everything. Weird that you have 2 copies of all of your DNA because we're trying to make 2 cells. So we're going to rip those chromosomes apart and move them away from one another. When we rip those apart, and this was in the metaphase picture that I had you draw, we rip them apart at their we rip apart chromatids from one another. We really call the whole thing at this stage, in this phase, at this moment in time, sister chromatids. When we rip them apart, we get to rip them into their individual halves. I know that it's confusing, and you're like, good gravy, we're using so many C words. I know. We'll go through that. Please don't fear. Okay? Chromatid, half a chromosome. I promise. I know the vocab is a lot. We're going to work with it. We're going to pull them to the opposite sides of the cell. They're going to move away from one another. Just link anaphase away in your brain. All right, next picture. Pause button, draw this picture. And we're going to telophase. And if you're thinking, oh my gosh, she's about to say telophase 2, nailed it. This is the point where we don't need those spindle fibers anymore, so they retract back into their centromeres, just like Spider-Man pulls his web back. That chromosome is going to relax a little bit. It is now no longer 6.4. We have 3.2 on the top and 3.2 on the bottom, so we can relax a little bit here. And our nuclear membrane is going to come and it's going to circle back around that chromosome because we want to make sure we're always protecting the DNA, except in those brief moments during metaphase and anaphase. So we'll redraw that guy. And then, pause button again, draw this picture. Last but not least, we are going to actually split those pieces apart. You can see the difference between an animal cell and a plant cell, how they do it, but we are dividing the cytoplasm. So cytokinesis, what it means is cyto is cytoplasm. Kinesis is like a knife. So we're going to knife through the cytoplasm. In an animal cell, that literally means we rip the two apart. In a plant cell, it means you drop a plate down the middle. The vesicles contain extracell membranes, and so they separate those two pieces. So we're going to make sure that we have two separate cells at this point, cytokinesis, knife, apart the cytoplasm. At the end of mitosis, this is kind of what the whole big picture looks like. So you can see as we went through interphase, G2, S phase, interphase containing G1, S, and G2, and then that pro, meta, ana, telophase through cytokinesis. And of course, giving a little shout out over here to those cells that never go through and they just do G0. Both of these cells will, upon receiving growth factor, go back through this process. So again, this is why you are half old and half new at all times, because you're always going through this process. Last thing to keep in mind here is that we are always producing two identical cells. They're going to have the exact same function. The one that it split in part is always because we are copying the one cell that we started with. So stomach cells only make stomach cells. Skin cells only make skin cells. Hair cells only make hair cells. Pancreatic cells only make more copies of pancreatic cells. Bladder cells only make more bladder cells. You get the picture. It has to be identical. You are not producing new random other stuff. We're making an identical copy. So what happens if that just like it all falls apart and that doesn't go properly? That's what we call cancer. Sometimes things just do not happen in the way that we are expecting them to, and so you will get cancer. I'm not going to take the time to play this video during this lecture. What I want you to do is pause button and go watch this video, or at the end of the lecture, know that you need to come back and watch this video. So what is a mutation? Remember, we've talked about this one already. So it is some part of the DNA that we have damaged in some way. There are a number of things that we could have done to damage your DNA. It could have just been bad from the start, like you got it from mom and dad and it wasn't good. You contracted a virus that damaged your DNA in some way. You smoked, or there was a chemical near you, or you inhaled something that wasn't supposed to be there. You sat in the sun for too long without your sunscreen on. Sometimes just this divisional process doesn't work properly. So a lot of things that could cause this stuff to have some sort of a change. There are two ways that cancer gets to you. And so if you want to hit the pause button, that would be smart here, and take a real look at this slide here. It's in your book as well. It's really nicely done in your book. This is just another version of it. I try to always give you like an alternate thing. So the normal cell, we're going to start on this side, so the left-hand side of the diagram. Normally in your body, you have two types of things. You have a thing called a tumor suppressor gene, and you have a thing called a proto-oncogene. If you're thinking, ooh, onca, that's a big fancy word. Yes, think of the study of cancer oncology. This is where the name comes from. They're called proto-oncogenes. And then tumor suppressor, it would stop something from happening. So that's literally what it does. Tumor suppressor genes stop the cell process from happening. There's a variety of reasons why they need to stop the cell cycle from going through. That's their job. Their sole function is to just disrupt things and say, nope, you're not going to go through. You're not going to do that. The proto-oncogene says, thumbs up, things are going great. We're going to go ahead and go through that divisional process. And when both of these are happening to a particular cell line, you end up with normal cells. So things are working just as they are supposed to. You can take a little peek over here at the brakes and the gas pedal on the car. So typically for a tumor suppressor gene, that would mean that your brake is working. If the brake works in your car, you hit the brake and you come to a stop. A tumor suppressor gene hits the brakes and it stops the cell from dividing. A proto-oncogene is like your accelerator. That's the gas pedal. So you press that bad boy down and you get to make more copies. You get to go faster. When those two genes get mutated, though, is when things get a little messy. So if we don't have the ability to stop bad things from happening, crashes can occur. Or cancer. And this cell cycle goes through at an abnormal rate, and we end up with cancerous cells. We get cells that are not supposed to be going through this divisional process that are instead going through this divisional process. And not those G0 cells. I mean, damaged or broken cells are going through this process when we intended for them to be sort of destroyed. A mutated proto-oncogene, a gene that is saying, hey, you should make a copy of this on demand, just one at a time as you're going. It's as though that gas pedal is just always down. You are just driving around town with that gas pedal down. You are not stopping at stoplights. You are not stopping at stop signs. You are swerving in and out of traffic. You have nothing but the gas pedal to the floor. That's it. That's all that you know how to do. It's almost like it just got jammed on and you are not in control anymore. And so you make cancerous cells. You go through this process when you were not supposed to. Your cell actually has 3 times. The cell cycle has 3 moments in time when you have these things called checkpoints when you are supposed to stop. They happen at the end of the G1 phase. We need to make sure that we have grown properly. If everything is all good, carry on to S phase. They kind of happen at the end of S and G2. If the DNA as it got replicated is too far damaged, your cell will do a couple things I'll talk about here. And it will not go through that meiotic process. So it will not go through prophase, metaphase, anaphase, telophase. Those things are just not going to happen. If you get into the middle of the mitosis phase because you have just skipped G1 and G2 because for some reason your accelerator is stuck, there's one more fail-safe that hopefully somebody notices that, like, wait a second, we have mistakes within us. And then you stop that process and you don't finish everything up and you destroy yourself in the middle. But it just, this cancer, these tumor suppressor genes and these proto-oncogenes don't listen to those checkpoints. They just go ahead and they just move on however they want to, or they don't stop things when they're supposed to. So a little more about each of those checkpoints. If you are curious about how those work, if you have more questions, bring those to class. Definitely, I'm happy to answer them for you. I am not a cancer biologist by any means, but I do think cancer is really fascinating. So if you have some questions, I'm happy to answer them from this molecular standpoint. Other kinds of questions about treatment and other things, not so much, but I'm pretty good on the genetics behind everything. These are the 2 things that will happen so this is like kind of the picture that's out here between a normal cell and a cancerous cell as they go through that division. Just kind of this is where tumors come from because you have that uncontrolled cell growth to get a mass. What you would hope a normal cell will do is it commits this thing called apoptosis. So if the cell will literally commit suicide in and of itself, and it'll say, hey, I am too far damaged. I am not going to be good for the body. If you let me continue, things are not going to go well. So it will commit suicide. Your immune system is also a failsafe between these two processes. If your cells, are not committing apoptosis, they will come through and attempt to commit murder. And they're going to make sure that everything gets taken care of. But sometimes these cancerous cells are very tricky, and they shut your immune system down, and they do not get murdered. They're really good at circumventing the system. They won't kill themselves, and they won't get killed by somebody else. They are so sneaky, and it's so tricky sometimes to find out that you have cancer because the rate of growth can be just slow enough, and those cancerous cells are avoiding your immune system in just the right way that's why some cancers don't get discovered until much later, and at that point where treatment is really difficult and you have a struggle with surviving, and that survival rate is really low. Cancers that grow really fast, or we have really great prevention programs, we know where and which cell lines to go ahead and go in and look for, and so then we can stop those guys in their tracks. We can boost your immune system a little bit there. So a couple ways on treating cancer. These are your general pieces for surgery, radiation, and chemotherapy. As you go do your cancer research, I am curious to see, do you have some drugs that you have noticed in the chemotherapy section? Are there some specific names of things that you can find specific to your type of cancer? Is there a new latest and greatest treatment that is out there? Obviously, surgery is pretty basic. Just cut it out, get rid of it. Radiation, just a low dose x-ray, literally same thing that you get. There are other high dose types of radiation that you can receive. What is specific to yours? How do we use your specific cancer treatment to get rid of that in a patient who might have that type of cancer for you? Great video just to leave you with here on the end. I'm not going to play it, but if you need to hear all of this again, if you would like just, I don't know, a couple days from now or right before the test, a quick recap on everything, this cell cycle video will do that for you. Same cartoon people that I really love, they have a really great cell cycle in cancer, just 10-minute recap on everything. If you have questions, please let me know, and I am happy to help in any way on anything you need.